putting them on as we go through them. All right, so we have a couple more research questions to deal with from chapter 4, and then we'll transition into chapter 5 and do our quiz. So thank you very much. So what's unique to plant cells? And I know we talked about what's unique to eukaryotic cells, but plant cells are interesting. Plant cells are very interesting in terms of structures that they have that you don't find in other eukaryotic cells. So here are some of those structures. Plant cells contain chloroplasts, and they are not the only eukaryotic organisms that contain chloroplasts. You find chloroplasts in green algae and some others, but it's just interesting. And so like mitochondria, chloroplasts have a double membrane, implying that they may actually have their origins outside of these organisms that are photosynthetic entirely. But inside of those membranes, you have the machinery for carrying out photosynthesis. Also, like mitochondria, chloroplasts contain their own DNA, and they can actually replicate separate from the cell, like mitochondria can. And again, a lot of this implies that possibly chloroplasts have their origins separate from the origins of the organisms that contain them. Although there are some big issues involved with that, many of which we'll actually talk about during our lecture break today. So save those thoughts. Like the mitochondria, again, a lot of similarities between chloroplasts and mitochondria. Like the mitochondria, there's a space inside of the inner membrane. We call that the matrix in mitochondria. Do you remember that term? I talked about on Friday. Inside of chloroplasts, we call that the stroma, this liquid inside of there. And within there, we've got these stacks. Grana are the stacks of thylakoids. And thylakoids are actually a third membrane that's just stacked up. And provides an enormous amount of surface area and contains the machinery, chlorophyll being part of that machinery, necessary to harness light energy. Another reason why I wanted to talk about this question, what's unique to plant cells, is I think it's a good reminder that there need to be some organisms that actually make their own food, right? Plants aren't making their own energy. They are harnessing sunlight. You actually can't make energy. It's impossible. But they are making their own food. And that's really important because it allows organisms that can't make their own food, like us, to actually live and to thrive. And you're like, what are you talking about? We can't make our own food, right? I put a Pop-Tart in the toaster this very morning, right? But again, the Pop-Tart brought you goodness from somewhere else, right? You did not make what was in there. And they're like, why use a Pop-Tart? That's a terrible example. They're disgusting. And you know what's actually cool is they're flammable once they get to a certain temperature. And so I don't know if you knew that or not. It's cool, but you have to be a little bit careful taking your Pop-Tart way past the point where it gets a little dark. You know, if you like your Pop-Tarts a little dark and toasty, just be careful because they actually are flammable. All right, some other structures that plant cells contain. Plant cells have to maintain water balance a little bit differently than a lot of eukaryotic cells do because they have such a high demand for water. And you're like, I have a high demand for water. But your demand for water is nothing near what a plant's demand for water is, because water is actually a reactant of photosynthesis, what plants use to make their own food. And so plants have a really high water demand, and because of that, they really have to regulate the amount of water. And so they've got this central vacuole inside of the cell that usually accounts for a very large percentage of the cell's volume, enclosed by this membrane called the tonoplast, and it just stores a lot of excess water and a lot of materials that tends to encourage water to move into it, keeping the solute concentration pretty high. So they store salts in there, they store sugars in there. Some plants that only go through part of the process of photosynthesis at night actually store intermediate products inside there. We'll talk a lot about that when we get down into the mechanics of photosynthesis. But actually really fantastic. It's a great storage reservoir. It's like having a house with a swimming pool in the backyard, but you don't really use the pool to swim in. You just use it to store all of your goodness, right? Like to store drinking water and to store, any food that you might need at any point. And it's just this massive reservoir. And you're like, why wouldn't I just use my pantry? Makes a lot more sense. And then I could swim in the swimming pool. I don't know. My analogy broke down, but I thought that sounded cool. Yeah, Rachel. Waste? Ooh, it should say waste products. Ooh, look at that R that snuck in there. That's kind of fun. I'll leave it. Yeah, it should say waste. But I'll leave it. That's kind of fun. Inside of there also you can contain enzymes and other materials for chemical defenses. You find this a lot in plants that are subject to high levels of herbivory, right? Where a lot of animals are just chewing on those materials. And then some plants that need to actually defend themselves just pack their central vacuole full of this really bad tasting or really dangerous chemical. And so cattle come in and graze on it and then leave with like a really bad stomach ache and will learn to not eat that plant in the future. It's kind of fun. Cell walls. And again, cell walls, these are not entirely unique to plant cells, but the way they build the cell walls is unique to plant cells. And so they're made of cellulose, which is a complex glucose polymer. We talked about that when we talked about our biomolecules. And they actually have two layers to their cell wall. Both layers made up of cellulose, but the primary cell wall is fairly flexible and soft. Secondary cell wall more robust. And then they can actually be further reinforced with lignin. We actually talked about this in organismic biology, and this is something that is unique to vascular plants that can actually now grow up because they have ways to transport water through their tissues. And if you're actually going to grow up, you need to be able to combat gravity pulling you down, right? So you infuse your cell walls with lignin, and it makes it really difficult to actually break down that plant material. And that's why, eating leaves is not energetically favorable. I don't know if you knew this or not, but if you actually do a good job chewing your food, it requires more energy to chew your lettuce than you access eating it, right? If you do a good job chewing your food, if you don't chew your food and you just swallow whatever goes in your mouth, you actually get more energy than it requires you to chew it because you didn't chew it. But for many people, you actually invest more energy than you get. So you're like, why are we eating lettuce? And I've got three answers to that. One, it's a wonderful vehicle for the dressing on your salad, right? And that's really what you want. It also makes you feel good about putting an enormous amount of meat onto your plate, right? Because you're like, it's not steak, it's a salad, right? I mean, yeah, it may have a 1.5 pound, piece of tri-tip sliced up on there, but it's a salad. And then second, it's high in fiber. It just keeps things moving regularly, which for you may not mean a whole lot, but as you start to get old, like I am, those kinds of things mean more. We'll have some conversations years from now when you'll come back and visit, and you'll be like, remember that time in class you talked about lettuce being high in fiber? I finally appreciate that. Thank you, Dr. Engel. You changed my life. At which point I will tell you you're welcome. All right. Yeah, and so these cell walls are an interesting structure because the cells need to communicate in very intimate ways. And so a lot of that communication has to do with cell to cell actually touching each other. And so there are structures that penetrate through the cell walls called plasmodesmata. They are cell junctions, the cell version or the plant version of a gap junction. And they actually move through cell walls to connect adjacent cells. It's a great way of keeping that level of cell communication high. Gap junctions do a wonderful job of that. And these are gap junctions in plant cells, plasmodesmata. All right, to show you a couple of images of what's going on. Here's the chloroplast. Here's your double membrane. And again, here's a third membrane layer that's forming just these really complex thylakoid discs stacked on top of each other. Enormous amount of surface area. Keep that in mind. Same thing is true in the mitochondria, that you have an enormous amount of surface area for lots of wonderful things. Here's an electron micrograph of chloroplast. You can see the granum, a single stack of thylakoid discs, and then plural would be grana throughout the chloroplast. Yeah.
How was an electron microscope picture if it's like cut in half?
Oh, well, that's fine. I mean, you just take a cross-section. You can do that with electron microscopy all the time. So you take a cross-section, and then you immediately plate that material with some kind of a gold alloy, and it just plates everything that's there. You do it really quickly so it doesn't deteriorate and just like completely fall apart. But yeah, you can do cross-sectional electron micrographs of lots of things. And you'll see this of like roundworms. And they're like, we call them roundworms because they have a circular cross-section. And they'll be like, you want to see? And they'll cut one in half and plate it with some kind of gold alloy and get you an electron micrograph of it. And you'll see that nice circular cross-section. But yeah, you can accomplish that.
So they cut it before they take the picture?
Yes. Yep, you bet. And actually, they put it in epoxy first, and then they slice that with like a deli slicer. It's another story for another time. So here are the plasmodesmata, these gaps, these gap junctions penetrating through the cell wall to connect the cytosols of adjacent cells. And then here, if you look at the cell wall, you can see primary cell wall and secondary cell wall, the middle lamella. Okay, just this really robust structure that really does a good job of resisting change. And the main job of the cell wall is to keep the cell from rupturing. Okay, because if you've got a lot of water moving into these plant cells to fuel photosynthesis, there's the risk of just having the cell swell so much it blows up. That's what happens in our body if you drink distilled water. Your blood solute level is going to drop so much that water is going to rush into your red blood cells and they'll start blowing up. That's why you don't drink distilled water. But plant cells don't do that because the cell wall keeps them from exploding. They would just put an enormous amount of pressure against the cell wall, but they wouldn't explode. That's bonus information. And then here's an electron micrograph of the cell wall showing you the primary, secondary, and the middle lamella. Yeah, Alyssa.
Do you have to know the names of all the parts of them?
No, I think it's just, it's good to know, like if you were asked, explain how the cell wall of a plant cell is unique, right? You've got some material that you could talk about. You could talk about the presence of cellulose, that complex glucose polymer. You could talk about that oftentimes it's infused with lignin to help, to overcome gravity. And then you can talk about the primary, secondary layers in the middle lamella, right? So you can talk about some of the unique features of the cell walls in plant cells. Does that make sense? All right. I think this is the second to last question. How does the space around, it may actually be the last question, how does the space around animal cells differ from other eukaryotes? So now kind of taking a step back and saying, okay, well, let's look at the cell in context, right? What's going on around it? What's in the surrounding environment? So here are some of the materials that you find in animal cells that you don't find in other cells. One, cell adhesion molecules. And so these are oftentimes glycoproteins. Glycoproteins are a combination of proteins and sugars. So two different biomolecules strung together. They're incredibly sticky and do a really good job of creating adhesion, which is why we call them adhesion molecules, of actually sticking the two cells together. And this forms what's called the extracellular matrix, are these glycoproteins sandwiching those cells in place. So you can't really think about the body of an animal as just these free-floating cells. I mean, all of these cells are very highly coordinated and are packed together in very sophisticated ways. And then another one is cell junctions that you don't find in other cells. I know we just talked about plasmodesmata, but you don't find those in animal cells. Here are some junctions that you do. You have what are called desmosomes. Desmosomes are a type of adhesion junction, holding cells together, holding them together really, really well, resisting a great deal of force that would tear them apart. And so one type of adhesion junction is called a desmosome, and it uses intermediate filaments to weld them together. Then you have another type of adhesion junction called adherens junctions, and these use actin. They use microfilaments to do the same thing. To, again, prevent the cells from being torn apart. So you find these in tissues that are prone to stretching. You find it a lot in muscle tissue, where the tissue as a whole is stretched, and you don't want those cells tearing apart. You find it in connective tissues as well, ligaments and tendons and fascia, to keep those cells from ripping apart. All right, make sense? And so this is what I was getting at with the quiz on Friday, where it said to provide a type of cell junction and then give the cytoskeletal element involved. These are your two best choices to go, because it's very clearly in desmosomes, intermediate filaments, in adherens junctions, actin, microfilaments. Another type of junction, tight junction. Tight junctions hold the cells so close together, fluid can't move in between them. So it's not necessarily keeping them from being torn apart, it's keeping them smacked together so tightly that fluid can't move in between the plasma membranes of those cells. And so you find these in places where you need to have a really good lining. You find them throughout the gut, in the lining of the stomach, the lining of the intestines. You find them in blood vessels, lymphatic ducts, anywhere where you want a really good lining and you don't want fluid leaving, right? You expect to find these tight junctions. They're not as good as adhesion junctions as keeping the cells from being torn apart. I don't know if you've ever done this, but you dissected an animal and you grab the intestines. You can actually rip them apart if you want to. It takes some force, but it's much easier to rip apart than to rip a muscle apart, right? You can rip the entire length of a muscle fiber away from the rest, but to actually rip in the middle of a fiber is really, really difficult. But you can rip, intestine apart. And this is like if you eat pig intestine or something like that because you want to, you find that too. You cook it and the junctions get even easier to deal with. Have any of you had pig intestine? It's really good if it's prepared well. You have to prepare it well, and you got to clean it really well, because think about what's in the pig intestine before you eat it, right? You don't want that flavor there, but if you can get that flavor away, it's actually really good. All right, last type of cell junction, gap junctions. Like the plasmodesmata, they connect cytosols of adjacent cells. And so this is wonderful. Then you don't have to rely on diffusion to get a signal to the neighboring cell, right? It doesn't need to leave your membrane, enter their membrane. Your cytosols are connected. And so this complex roadway for transporting materials can just transport materials from one cytosol to the next. Yeah, Joey.
What kind of a junction do nerve cells use?
Nerve cells it's not a type of cell junction, it's called a synapse. It's a space in between the cells. The cells actually aren't physically connected together. They're bundled together in a nerve, but they're not physically connected together. There's actually a space in between them. Okay. And so one neuron will dump material into that space. The other neuron will bind that material and execute some response to it.
Okay.
Yeah, that's a good question. All right, and then lastly, the extracellular matrix in animal cells. And so, most of the animal cells are locked into this extracellular matrix, again, made up of glycoproteins, just smashed and stuck in place. And then depending on the tissue, this extracellular matrix can be further hardened. In bone, in cartilage, you harden that matrix even more by infusing it with minerals. And so not only does it lock the cells in place, but then it becomes very hard and rigid and actually forms some nice protective structures. In other cells, it's just, again, made primarily for holding the whole cell together and forming the bulk of the tissues. A lot of this is an adhesion, just holding stuff together, but a lot of it is also controlling whether or not the cells divide. Cells won't typically divide if they're locked in a matrix, right? They're locked in a matrix, there's really nowhere to go, right? I mean, if they start dividing, it's just going to cramp the space that they're given within that matrix. And so a lot of this is a way to keep cells from dividing when they ought not to be, And a lot of it is also for controlling developmental processes. When you need a lot of cell division happening, when you need to shut it down, when you need to turn it back on, a lot of that is executed by the extracellular matrix. Yeah, Joey?
So, like when we're thinking about like skin, bones, and tendons, is it almost better to think about like the cells that are there as sort of like they're just kind of regulating that tissue, that like extracellular
Yeah, the
matrix?
extracellular matrix is doing most of the work that you associate with the tissue. The cells maintain the matrix.
Okay.
Yep, yeah, it's a good question. Yeah, so in bone, in cartilage, in connective tissue, other connective tissues like cartilage. I guess we already talked about cartilage like tendons and ligaments and fascia within muscle cells. Yeah, the matrix is doing most of the work you associate with that tissue. The cells are maintaining that matrix. All right. So here are a couple of images, actually several images. Here are all three cell junctions modeled in a single image. And so here you have adhesion junctions. No, sorry, these are tight junctions. So here you have tight junctions holding the membranes so close together, fluid can't move in between. Here you have adhesion junctions, and so these would either be desmosomes if it's intermediate filaments involved, or adherent junctions if it's actin, microfilaments involved. And then here you have some gap junctions connecting the cytoplasms. Here's a zoomed in picture of a adhesion junction or anchoring junction, and then an electron micrograph showing where you have these cytoskeletal fibers holding these cells together, again resisting them being torn apart. And gap junction attaching the cytosols of two adjacent cells, and then an electron micrograph showing where these gap junctions are, not really showing you the detail of those gap junctions, but showing you where they are. And then here's your extracellular matrix. There's just composition or compilation of glycoproteins forming this sticky material, sticking the cell in place. All right? Any questions on this material before we transition to our quiz? All right. Thank you all for your your smiles and your chuckles. And if you could kindly put away everything except for the little slip of paper. Does everyone have a slip of paper? A little slip get to everybody? Make sure your name makes it on that slip of paper, because some of you are emailing me pictures from phone numbers. So just make sure your name ends up on that slip of paper. Okay. Number one, what are the two most common lipids in cell membranes? Number two, what is a material that won't or that wouldn't move well through a lipid bilayer? And then number three, name two functions of cell membranes. Absolutely. About 30 more seconds. All right. If you haven't already done so, wrap up your thoughts, snap a picture of your quiz, and go ahead and send it in. There's no need to be sorry. I'll enjoy this when I look back on this months from now and see all these quizzes in between pictures of my children.
I can just decide to send whatever subject I want. Title just to make you laugh.
Oh, is the subject of the email?
I'm just always trying to make you laugh. That's it.
It's not hard. I'm a pretty jovial person, you know. All right.
It's not funny.
Everybody send in their quiz. All right, so what are the two most common lipids in cell membranes? Phospholipids and cholesterols. Or you could give two varieties of phospholipids if you wanted to, because phospholipids are more common than cholesterols entirely. But you could do like phosphatidylinositol, phosphatidylcholine. Yeah.
What about fatty acids, like the tails in the phospholipids?
Oh, sure. Yeah. Yeah. So you said phospholipids and fatty acids
I said fatty acids and cholesterol.
and cholesterol. I see what you did there. I like it. What is a material that wouldn't move well through a lipid bilayer? I mean, you could do anything that's polar or charged would not move well through a lipid bilayer. So you could put all sorts of different things there. Name two functions of cell membranes, controlling what goes in and what comes out of the cell, acting as a barrier, also protecting the internal compartments of the cell, regulating cell communication, all sorts of different functions carried out by cell membranes. All right. So we're going to do our lecture break now, and I know it's a little weird because we just had a quiz and we just had a lecture break. But what I want you to do is think back on our discussion of mitochondria. Think about our discussion of chloroplasts, where there's evidence, there are evidences that chloroplasts and mitochondria have a separate ancestry than the cells that they are inside, right? They have their own DNA. They divide separately from the cell. They don't have a single membrane surrounding them. They've got a double membrane. In the case of chloroplasts, 3, sometimes even 4 membranes. And so there are evidences that those organelles don't originate inside of those cells, that they weren't originally part of them and actually have a separate ancestry. What I want you to do is think about what would be required if that's true, right? What would need to happen if that were true? If there were cells that lacked mitochondria and then, ate something that became the mitochondrion, okay? So I want you to, with your group, With your discussion group, there should be 5 or more of you. Were any of you not here on Friday and you don't have a discussion group? Because I'll put you in one. Okay, we're all good? Cool. All right, so what would have to happen mechanistically to make that happen? What steps would need to take place? Go ahead and talk about that for a couple of minutes with those in your discussion group. Come up with, try to get at least one thing that would need to happen mechanistically in order to make that possible. Okay, to where mitochondria and chloroplasts could have a separate ancestry than the cell, which basically means that at some point they need to enter into the cell, right? So think about some mechanics that would need to happen. And this will be a good discussion for us to begin and to continue throughout our semester. So go ahead and take a couple of minutes starting now. There you are. I was wondering if you had to leave. Are you feeling okay, Mom? You're doing okay.
And then Alyssa, you can say what you said.
What were you saying about they would have to have some other source of energy?
Well, the original more complex cell that would ingest other smaller cells would not have mitochondria in it, and the energy needed to be a bigger cell. So basically, you have like 2 small cells, one ingesting the other. Larger size, but like you have a more complex size than a mitochondria.
There would have to be, what would be the cell's intention of digestion? What's the,
Right, you have to ingest it, like, oh, just kidding, I'm not gonna ingest it. Yeah.
So there would have to be a malfunction.
Right, so it has to, in the mutation,
All right. We're going to go clockwise this time. On Friday, we went counterclockwise. We started here and went this way. We're going to start here. And if you don't have an idea that you want to share with the class, I guess you could say pass. But yeah, we'll see. You have some ideas on what would need to happen mechanistically for this to work, to that at some point you have mitochondria or some organism enter into the cell and be incorporated as mitochondria or alternatively chloroplasts.
We said that the cell would need to have a separate energy source pre-existent to the mitochondria.
Okay, good, yeah. And so mitochondria tend to be the place where you get the bulk of your energy conversion, right? We'll talk about this a great deal in a few weeks. It's not the only place, and we even mentioned this when I talked about the mitochondria, but part of cellular respiration actually takes place in the cytosol, but it's very energetically inefficient. So having mitochondria makes that more energetically efficient. So yeah, that's something. Like you have to have a means by which you convert whatever it is you're eating, chemical energy, into chemical energy that then you can use. Good. You have some ideas, group 2? This is group 2, right? 1, 2, and then 3, 4, 5, 6. Group 2. Are we going to take a pass?
We're not sure because we didn't really cover it, but we kind of thought of phagocytosis.
Yeah, so you need some mechanism by which you can incorporate a large piece of material. Because that's really interesting. We're not talking about just eating dissolved organic material here, which a lot of our heterotrophic organisms do, right? They just eat dissolved organic material, and that can enter in by diffusion, or they can do what's called pinocytosis and just kind of drink some of the material around them. We'll talk about all of these mechanisms in chapter 5, which we're talking about this week. But yeah, you have to have a way of bringing in something enormous into the cell. And that's interesting, right? That process is highly controlled. When we look at phagocytosis, it's a highly controlled process and actually involves binding what you're going to eat first usually. It's usually what's called receptor-mediated endocytosis, but we'll talk about that. Group 3. We have some added thoughts.
You would have to have additional machinery with which to communicate
Right.
between the foreign substance and the host cell.
Absolutely. Yeah, I mean, undoubtedly there is communication that takes place between the mitochondria and the cell, right? So you either need to already have that in place or that's something that has to develop, right? And then you have to figure out where you can read its signal saying, don't eat me, please, I'll serve you, right? It's just, yeah, it's very interesting. Group 4.
Thought of a few things.
Okay.
What I felt was most interesting, though, was that there had to be a way for this mitochondria to reproduce itself,
Uh-huh.
still be part of the cell, which
Right.
might imply that it needed to somehow get coding into the nucleus,
Yes.
so that the cell can reproduce more mitochondria when it divides and divides and divides and divides.
Sure. Yes, because they can divide independently, but that process is still controlled by the cell. The cell controls how many mitochondria there ought to be, typically. And so there's an enormous amount of communication that takes place between the cell and the mitochondria.
So, does the code for making a mitochondria in the nucleus of the cell, is it the same thing as other organelles? And then does the mitochondria reproduce separately?
Oh, yeah, that's it. That's a good question. So let's put it this way. You tend to reproduce the entire endomembrane system when the cell divides. Okay, so when the cell divides, the old endomembrane system deteriorates when the nucleus deteriorates. And then after the cell divides, as part of that division, you rebuild the nucleus and you also rebuild the rest of the endomembrane system, the endoplasmic reticula, the Golgi apparatus. But the mitochondria, that division is not as highly controlled. That it tends to just separate the mitochondria that are present in the cell into the two different cells. And so the mitochondrial division is separate from the cell division. But it has to be highly controlled to control the amount of mitochondria that are present. All right, group 5.
So your question was about the mechanism, right?
Yes, just some mechanism that would need to be in place.
So, well, everyone's been adding their good and bit on it. I think just what's the mechanism which allows this to happen is, it's coming from fraternal, right? It's fraternal DNA, right?
Yes, yeah, the mitochondria come primarily from your mother's cells.
And so, yeah, so it's from the ovum. It doesn't matter, from what guy. It's dependent.
Right, yeah, the sperm tend to not contribute any mitochondria.
So what you just last stated was vision of it is separated. Is that that's the whole meaning?
Yeah, I was talking about like a cell within your body. Not necessarily like where does it come from in the first place. And then once the ovum starts to divide, at that point it would be a zygote, it wouldn't be an ovum. And so when it starts to divide, then you still have to control that process of making sure each daughter cell gets mitochondria. Yeah. So you have to have some means by which you can control the organization. Yeah. So the fertilization is not cell division. So cell division happens following fertilization, but they're separate events. Yeah. Group 6. Yeah, Alyssa.
So they would need to absorb it, like ingest it without
Uh-huh.
digesting it.
Right, so you need some mechanism to prevent it from digesting that material. Which if it has, what was it, group 2 that you talked about phagocytosis? Like you'd have to have the mechanism by which you could bring in something really large, which is interesting because if you have that mechanism, right, you think you're bringing it in so you can eat it, right? Because that's an energetically costly event, bringing something that large into your cell. And now not only do you need the mechanism to do that, but you need a separate mechanism to tell you not to eat it, not to digest it. You've already ate it, but not to digest it and allow it to keep living. It's very interesting. And then the question is, well, why doesn't this happen more often? Right? And you could argue, well, maybe it did happen in several lineages of eukaryotes, or at pre-eukaryotic cells, but why don't we see this happening now? Which is a very intriguing question. Yeah, Charles.
That was actually what I was going to ask. Is there a way to maybe observe it now? Is it happening now? Or even force it to happen? Replicate it?
It's interesting. There are organisms that will like incorporate photosynthetic organisms into their tissues and not kill them. So you'll find examples of like jellies that you can actually watch them incorporate diatoms and dinoflagellates, photosynthetic organisms, into their tissues and not eat them, but not into a cell.
Yeah.
Right? Into the tissue and not eat it, that's one thing. Into a cell and not eat it, that's something entirely different. So, yeah, that's a good question. Yeah, Alyssa.
Oh, another thing was the cell ingesting would have to be more complicated and much larger
Sure.
than the one it's ingesting.
Yep.
But without any energy, without mitochondria to help it provide that energy.
Right, to make it an efficient. Yeah, and that's okay. There are a number of parasitic animals that either don't have mitochondria or they don't use them. And they're energetically inefficient, but they have such a reliable food source that it doesn't matter. Right? They live inside their food source. So yeah, I mean there are ways in which you can do that, but that is another issue altogether. All right, so our first 4 questions we need to deal with. What are the lipid components of the cell membrane? This is addressing our first quiz question. How do other biomolecules function in membranes? Carbohydrates, proteins. What is the structure of membranes? And how does the membrane function? Again, kind of just this introductory idea to chapter 5. Wednesday's quiz will be a little bit more focused and a little bit more detailed, but still covering the same idea and the same material. So what are the lipid components of the membrane? So we already mentioned this, made primarily of phospholipids and sterols, cholesterol being the main sterol that you find inside of cell membranes. And so these are 2 wildly different lipids. We even talked about that when we talked about lipids as a biomolecule. There's just an enormous amount of variation among the lipids. But what is something that unites all lipids, regardless of what they look like and how they're built? Yeah.
They have a chain of 3 carbons, a glycerol.
Many of them do. Yeah, phospholipids do. Your triglycerides do. Not all of them do. Many of the lipids do have a glycerol backbone.
And they have fatty acids.
Many of them do. Not all of them do. But they are all nonpolar. They are all nonpolar, meaning they fear water. They are all hydrophobic. And so phospholipids, they are polar and nonpolar. And so phospholipids are really interesting in that way. They are both polar and nonpolar. So they are both water-fearing and water-loving. So they have a portion, the head group, where the phosphate is, that's polar and water-loving, interacts well with water. And then they have these fatty acid tails that are water-fearing, that are nonpolar. And so that's a really interesting structure to an organic molecule. And so what you get is inside of water, these phospholipids self-assemble because they are both polar and nonpolar. And actually anything that's both polar and nonpolar will self-assemble into a ball within water. So you find that with detergents as well. Yeah, let me get Sadie and then I'll come back here, Alyssa. Sadie.
Are soap molecules formatted after phospholipids? Like, could they use that as the model? Where it's
For what molecules, I'm sorry?
phospholipids, so like one side of the molecule interacts with
Uh-huh.
oil and the other interacts with water.
So are what modeled after that? Soap. Yeah, so detergents accomplish a very similar thing. And many detergents are natural components. They're just harnessed from living tissues. Soap used to be made entirely from rendered fat. You would just render animal fat and you could kind of get that consistency where you get polar and nonpolar materials. But yeah, synthetic detergents are made that way as well and are modeled in that way. Yeah, listen.
Is that why soap has bubbles?
That's why soap forms bubbles. Yeah, and why you can actually blow bubbles and you'll form this circular object with water trapped on either side of that layer. And then when it pops, you get a little bit of water. But yeah, I mean, anything that's both polar and nonpolar is going to self-assemble into circular objects within water. Now, at low temperatures, this becomes semi-solid. And what we mean by semi-solid is that it's solid, like a solid object, having a constant volume and constant shape, but there's some flexibility within there. Things can move around within it, but it maintains a constant volume and a constant shape. And then as you heat membranes up, they get far more fluid-like, behave a lot more like liquids with a constant volume, but a variable shape. Variable shape. So sterols, like phospholipids, are polar and nonpolar. They're smaller, though. The polar area is just a hydroxyl group. And then the nonpolar is this big, complex carbon and hydrogen ring. But again, they are polar and nonpolar. So this polar portion of the sterol, would it be cholesterol or something else, it's going to interact well with the water outside of the membrane and inside. And then the nonpolar region is going to interact well with the fatty acid tails. So again, a range similar to phospholipids and will self-assemble in water. Mainly cholesterol in animal cells. And this is what the sterols, and primarily cholesterol in animal cells, are what help keep the membrane from becoming solid. And what might some of the consequences of having a solid membrane be? So think about if the membrane, this structure surrounding the cell, if that were solid, what are some of the consequences? Yeah, listen.
It would be harder to transfer things in and out.
Absolutely. Yeah, I mean, it's hard to move objects across a solid barrier.
Also, it'd be harder to divide.
Absolutely. Yeah, absolutely. Daniel. Yeah. If you got to move, so now you're actively pumping materials, right? Now this gets really interesting, and you're like, well, you can pump stuff through a solid object. Yeah, but you got to have some opening within it. Yeah. So, any other consequences that you can think of having a solid, membrane? Yep, absolutely. It'd be very hard for those cells to move, and that would be a big one. That'd be a big issue if the cells had no freedom to move or to flex. And it would create some issues with the extracellular matrix as well, and we'll get there in a little bit. So here's a phospholipid. Here's our polar or our hydrophilic end. Here's our phosphate group. And then here are our fatty acid tails forming the hydrophobic or the nonpolar. And so here is the spontaneous membrane that will form in water. So what you'll get is you'll get two layers of these phospholipids forming in such a way that the polar head can interact with the outside of the cell where there's water and with the inside of this membrane where there's water. It's not necessarily a cell, it may just be a membrane. And then the two fatty acid tails will interact with two fatty acid tails from the other phospholipid, and you form this massive nonpolar region. And that's why in the quiz, when it asks you what kinds of materials would not move easily through a lipid bilayer, anything polar is not going to move well through this because there's just this massive water-hating, and it's not just water, anything polar, right? Water gets the bad rap because water is the most common of all of the polar materials, but anything polar is not going to interact well in this massive hydrophobic area. And so here's what it would look like if it were completely solid. Notice the fatty acid tails are not kind of waving, they're not moving around at all. And so now you have something where it used to be really hard to get material through it, and now it's virtually impossible to get material through it. On top of that, the cell is very static and can't move and is very inflexible. And so here's showing you the membrane with our cholesterol sprinkled in. Told you that cholesterols are oriented the same way as the phospholipids in a bilayer with the polar portion facing out along with the head of the phospholipids and the nonpolar region mingling or swimming among the fatty acids from the phospholipids. And again, cholesterol functioning primarily to keep the membrane fluid, keep the membrane from solidifying. Yeah.
How come it's a lot easier for non-polar things to get through when there's still a hydrophilic head?
Oh yeah, that's a good question. I mean, they're basically running away from the water in this solution. right? So if they find something that where they can actually escape that, a lot of times they'll get stuck inside of here, but it'll start to disrupt the way the membrane works, and they'll get forced out, and then they'll get forced into a place where there's less of them.
But wouldn't the heads like make it want to go away?
Yeah, but I mean, the interaction with the head is temporary, because as soon as you get through that wall, and again, you have to remember this is fluid, right? So these head groups are moving around. Once you get through there, there's not that same repulsion. But yes, yeah. I mean, polar materials are not going to interact well with the head groups, but they're also not interacting well with everything that's outside here, right? It's a polar environment that they're not interacting well with anyways. And so they're going to end up in here where they can, and what we say with interact well, it all has to do with the ability to make bonds. Because when they make bonds, they form something more stable than what you had by itself, right? Otherwise, the bonds don't form. And so when we say interact well, it's the ability to make bonds with it. So, yeah, that's a good question, though. All right. We're going to go ahead and end here. We've got we're about a minute and a half before our scheduled end time, but the next slide is introducing the next question. And so we'll actually go ahead and end here. We'll pick up here on Wednesday. Have a wonderful day. Enjoy the rest of your Monday. And I'll put today's lesson up on YouTube when I have time.